Hello everyone. As we all know, the flavoring agent which causes a sour taste in many of the prepared foods is none other than vinegar. So let's get into the depth detailing of vinegar production. The word vinegar is derived from the French word vin, which means wine, and aigre, which means sour. So basically, vinegar consists of acetic acid, which is around 5 to 20%, along with water and different amounts of fixed food acids. The coloring matter is present, as well as salts and a few other fermentation products are also present, due to which flavor and aroma are imparted to the vinegar. Now, if we go by the definition, vinegar is a liquid which is fit for human consumption, and it is being produced from a suitable raw material of plant origin which contains starch, sugar, or starch and sugars, and is made by the process of double fermentation, which is first alcoholic and the later stage is acidulous. That means formation of acetic acid. Now we will discuss about vinegar production. The production of vinegar involves two conditions, anaerobic as well as aerobic. In this, first, the glucose which is obtained from the raw material is being converted into ethanol by yeast, and the ethanol, this process is called alcohol fermentation, and later this ethanol under aerobic conditions is converted into acetic acid, which is the vinegar, in the presence of acetic acid bacteria, or we commonly called it as vinegar bacteria. And the acetic acid bacteria belongs to the member of genus Acetobacter. Why we use this bacteria? Because this bacteria has the characteristics or the ability to convert ethyl alcohol into acetic acid. So this is the diagrammatic presentation of the vinegar production. Suppose as a raw material we have selected apple, and this apple is alcoholically fermented by the addition of yeast, the apple juice. Then after the formation of ethanol, there is addition of acetic acid bacteria, that is Acetobacter, which leads to acid fermentation under aerobic conditions, and acetic acid or vinegar is produced, which is sterilized and preserved for further use. Now we will get into the detail of vinegar production. So the first step in vinegar production is the use of substrate. So the one of the critical steps in vinegar production is how to prepare raw material. So in this step, we have to obtain the fermentable sugar and juice solution, which has to be acidified. This process of acidification differs, which will depend which raw material is being used. विनेगर की प्रोडक्शन के लिए जो रॉ मटेरियल्स यूज किए जाते हैं, दैट इंक्लूड राइस, ग्रेप्स, malt, apple, honey, potatoes, whey, or any other sugary or starchy food. Now, the second step is involvement of microorganisms. First, we use the substrate in the form of raw material, which could be of any plant origin. Now, after raw material preparation, the alcoholic fermentation and acidification process plays a very important role in vinegar production. Now, there are two microorganisms which are involved The first is yeast and the second one is acetic acid bacteria. First, we will discuss about yeast. So the genus which we uses for vinegar production is Saccharomyces, which is a very common yeast used in beverage industry due to its high capacity to ferment sugar, which allowed them to colonize sugar-rich media in comparison to other yeasts, which are not tolerant to alcohols. So the tolerance to alcohol is a characteristic feature of Saccharomyces. That's why this genus is being selected. Now the common species which we use are Saccharomyces cerevisiae and Saccharomyces baianus for vinegar production. The second microorganism which we used for vinegar production is acetic acid bacteria. Now for the acetous fermentation, that is the conversion of ethanol into acetic acid, is being carried out by acetic acid bacteria, or commonly we call it vinegar bacteria, which belongs to the family Acetobacteriaceae, and the genus which we commonly use are Acetobacter and Gluconobacter. Now, there are three important production methods of vinegar, which could range from traditional to the latest modern methods, which we will discuss in detail. So, first one is Orleans process. This is also called as French process. Now, this method was made famous by the French and was named after a small town in France named Orleans. And Orleans is commonly, or we could say it is also known as City of Vinegars. So the name Orleans is derived from that town. Now, this is the one of the older and slower method of creating a high quality vinegar. We can call it as a traditional method. And in this method, the wooden casks are employed for the production of vinegar. Here you can see the diagram of wooden cask. It looks like somewhat like this. Now we will discuss the process of Orleans process in detail. This method involves the fermentation of vinegar inside a container, which is called cotton wooden cask. The container has holes drilled into it so that oxygen is allowed, because the second process of vinegar production requires aerobic conditions, as we have discussed. So to allow oxygen inside, there are drills. Now these holes also contain screen filters so that they prevent insects and other bugs from entering the cask. So they are covered. Now oxygen is necessary for vinegar production, as the bacteria that turns the solution into vinegar requires oxygen. Now to create the vinegar, the alcoholic liquid which is being produced by yeast is poured into these casks, and then about 20% of fresh vinegar is poured into the cask so that the process of fermentation begins. Once the Acetobacter bacteria oxidizes the acetic acid, the vinegar production is finished. Now, as you can see, there is a plug on the container, that is the wooden cask, through which we can collect the finished product, that is acetic acid or vinegar, and it is bottled for future use. So, Orleans process is the older or traditional method. The second one is generator process. This is a modern method and it utilizes the surface culture. Now, in comparison to Orleans method, which was rather a very slow process, many people tried to increase the production by implementing a new method, and the new method is generator process. In generator process, the production of vinegar involves spraying alcoholic substrate in the top layer of the fermentation chamber, where it is filled with material that carries a slime made of acetic acid bacteria, so that the acetobacter could react with the substrate and create vinegar. Here the substrate is ethanol. So ethanol has to be converted into acetic acid. So due to the heat that is made during the fermentation of vinegar, air is forced through the chamber to keep it cool. And the vinegar produced is recirculated 2 to 3 times until the desired concentration of vinegar is achieved. So this is about the generator process. And the third one is submerged fermentation. This is the most common method which is being used by the manufacturers for large scale commercial production of vinegar. Here it involves the submerged culture. Now we will discuss this in detail. This is the newer, more faster and more efficient method of vinegar production. As we discussed, it is used on large scale when the requirement is at a larger scale. In this method, there is a high-speed motor brake which breaks down the air which is being brought inside the stainless steel tank into tiny bubbles and is forced in the solution of alcoholic liquid so that the acetobacter can fasten the process of oxidation, that is oxidation of ethanol to acetic acid. And the final steps involve are filtration. The final product, that is acetic acid, is filtered and pasteurized so that it can prevent the bacterial growth and stop the enzymatic reactions, as the whole process takes only 1 to 2 days. So this method is mainly used by big industries. Now we will discuss about the various types of vinegar. These are, since we have already discussed that we use the different raw materials for the production of vinegars. So on the basis of different raw materials, these are the various categories or types of vinegars. That is balsamic vinegar, cane vinegar, as you can say it is derived from sugar cane. Then there is champagne vinegar, cider from apple, coconut vinegar, it has very low acidity. Then we have distilled vinegar. The raw vinegar is distilled. Then there is malt vinegar, as the name implement, derived from barley. Then there is rice wine vinegar. Then this vinegar is also very common, sherry vinegar. It is kept under high heat of the sun in wooden barrels. And then there is spirit vinegar. In this, the acetic acid, that is vinegar, is diluted with small amount of ethyl alcohol. So it is also called distilled vinegar or grain vinegar. Then there is white vinegar and grape vinegar. So all these are the different types of vinegar. Now let's have a look at the uses of vinegar. As we all know, vinegar is basically used in food products for centuries. Now vinegar, what it does, it retards the microbial growth and contributes sensory properties to a number of foods. It enhances the taste of the food products. Vinegar is also used as a medicine, corrosive agent. As we all know, in pickles, vinegar is a main ingredient. Sometimes it is directly consumed, although in diluted form, as a beverage for some ailments. In the food industry, vinegar is usually made as acidulant and in food. So along with this, it is also used in salad dressings, in mayonnaise, mustard, ketchup, bread, bakery products.
It starts with the right mix of apples. Some varieties are sweet and others are tart. This mix of flavors will spice things up. The apples float to the surface of a water-filled pit and drift onto a conveyor that takes them into the cider mill. Inside the mill, the apples spill into a washing station. Water sprays the apples as they bounce across rolling brushes. Next, the apples take a trip up to a grinder. Inside, a steel arm pushes the apples against a grater. It shreds them, pits and all, into a pulp. This pulp is known as pomace. The pomace lands in a funnel. From here, a pump moves it through pipes across the plant, where it arrives at a press. The apple pomace flows between two porous belts that wind around rollers. The rollers apply pressure to squeeze out the juice. The juice drips through holes in the belts. The process strains out most of the pulp, but some remains as the juice spills from the press into large catch pans. Then they pump this chunky apple liquid into a holding tank. From the tank, it flows through a revolving drum that filters out more pulp, pits, and apple stems. The fluid seeps through the perforations in the drum and flows into a large trough. After one more screening, only fine bits of apple and sediment remain. This gives it the desired cider consistency and flavor. The cider now flows into channels between hot steel plates. The heat destroys pathogens and bacteria that would convert the natural sugars to alcohol. This ensures that the cider is both safe to drink and non-alcoholic. It's now ready for bottling. A worker slides plastic jugs into position under nozzles. The nozzles descend and pump the boiling hot apple cider into the jugs. They're filled to the brim so air can't get inside. Air causes the cider to deteriorate. Next, the conveyor moves the jugs forward to a capping station. Plastic caps funnel toward the jugs as they approach. The caps land on the rims with perfect timing. Spinning wheels tighten the caps as the jugs ride by, hermetically sealing the hot apple cider, giving it a shelf life of approximately a year. The jugs then head into a tunnel where sprayers douse them with cold water. Next, machinery pulls the liner off of adhesive-backed labels. An electric eye detects the approaching jugs, cueing rollers to apply the labels. The filtered out pomace from earlier is cooked 4 to 5 hours. This caramelizes the sugars in the apples, turning the pomace into a highly concentrated spread known as apple butter. They bottle it while it's piping hot to preserve it. Although not technically butter, this fruit spread has a similar consistency.